All right, so in this lecture, we're going to start looking at carbohydrates. So this is going to be the introduction to carbohydrates, focusing mostly on overall structural characteristics. Okay, so it's actually easier, can be easier. Excuse me, it can be easier to go back to Fischer projections, which may have just made everyone nauseous. Sorry. So Fischer projections are these like bow tie structures where I'm like, forget the bow tie, just, learn how to prioritize things as needed. But the Fischer projections are useful for carbohydrates because we have so many adjacent stereocenters. So in general, what we do is we start out with an aldehyde or an OH that then goes into a ketone. And then we have 4 stereocenters in a row. And so the Fischer projection is helpful because what we can do is just remember whether or not the OHs are on the left and the right. And I just remember it goes, everything's on the right except the second one, which is on the left for the molecule glucose. So this would be the dextrorotatory glucose as a Fischer projection. Now, it turns out that if you have D-glucose, it doesn't sit around in this form too often. It will ring close in really, I think in most states it just sort of ring closes, but in aqueous solution, it exists as a chair structure form that results from this oxygen. When I say this, I should clarify with the structure. Excuse me. I'm going to draw my 4 stereocenters, but this is going to be an O that's now adducted to the C. And instead of having a CHO, I'm going to have a COH. So those two are now bonded with each other. And otherwise, it's the same. Now, as I alluded to, it's just easier to draw this as a ring closed form. Okay, so this exists in about 90. We get about what looks like about greater than 99% in the closed ring form. Okay, so what does that look like? So three-dimensionally, chair structures are better. Chair structures are better. Where we consider a chair structure where there's an oxygen inside the ring. Now, if I zoom back out, you can see that that oxygen that's inside the ring was the oxygen that attacked the carbonyl. Now what I'm going to do is, the great thing about glucose is all of these substituents are equatorial, except for, and I'm going to show it as a wedge right now, except for that substituent that was once an aldehyde. Was an aldehyde. I'm going to show that as an OH. Now I'm going to match that OH with this OH here. Okay, so that allows us to define the anomeric carbon and anomerism. Okay, so the anomeric carbon refers to the hemiacetal or acetal carbon, whether that's in a monosaccharide, a single sugar unit, or a polysaccharide, the polymer of a bunch of monosaccharides. So the hemiacetal, that is this carbon with an OH and an OR group. It's very weird because it's a carbon with 2 single bonds to oxygens without any carbonyl stabilization. Okay. The acetal differs very subtly from the hemiacetal in that it's got 2 OR groups. If you recall from Org 2, we called the hemiacetal, we said that refers to the half acetal because we're halfway there for forming the product. Now the anomeric carbon equilibrates to be up or down. And that is the stereocenter at that position. That's kind of a, it's a weird statement to write, referring to the stereocenter. Okay. So what I mean here is I'm going to draw two chair structures. And what I'm going to do is tuck that oxygen into the upper right corner for both chair structures. Now, these aren't chair flipped structures, if you're thinking about that. These are just the same chair structure. Yeah, you could flip everything. It's not that helpful because then a bunch of stuff goes axial, which isn't great. Okay, so for glucose, in both cases, everything is equatorial. Now, I want you to notice about the upper left, we go to a CH2OH, CH2OH. Okay, now the carbon I'm showing here is this is the carbon that's going to have a single bond to an O that's part of the ring, and then another single bond that's to an OH. Or it could be to an OR, which then connects to the rest of a chain on a growing polysaccharide. Okay, now what we could do is we could take the last OH, I'm going to keep it blue, and we could position it axial or we could position it equatorial. And both structures exist. It turns out that because these things can equilibrate, they can interconvert. What happens is we pop open the ring to get the open form, and then we close the ring to get the closed form. But in that opening to closing state, we can actually get to either one of these isomers. Now they're isomers, they're actually different types of isomers. These are called anomers of each other. Anomers and anamerism. These are, isomers with different stereochemistry at the anomeric positions. And stereochemistry is a vague word. I could have said configuration or wedge dash. Now, with chair structures, we don't actually need wedge dash. We have axial and equatorial to get us there. Now, when we talked about chair flipping in organic 1, we flipped all of the wedges and dashes, all of the axials and equatorials around. Here, we're not doing a chair flip. We're ring opening, closing, making a stereocenter with the opposite configuration. Okay, so these two things are diastereomers of each other. That is, we flip some, but not all. These aren't chair flip structures. Okay, now we can classify these anomers as being the alpha anomer, and I remember that because the OH is axial, and alpha is the Greek letter for A. So that's the alpha anomer versus the beta anomer. I always like the beta anomer better because it's equatorial. Everybody loves equatorial substituents, right? So it's a little bit weird that it's the second one, it's the beta, but alpha for axial. Okay, now if we look at percentages, we said that we had greater than 99% was the closed form. Now, which one's dominant? Well, it turns out that of that 99%, greater than 99%, we're at 37.3% of the alpha form, and then 62.6% coming from likely the stabilization of the equatorial substituent, amongst some other effects, sure. Okay, so we get a greater percentage of the beta, and then if you do the math, you're missing about 0.002%, and that comes from the open chain form, which barely exists at all, but allows us to interconvert between these two and establish an equilibrium. Okay, so glucose is an example of a glucopyranose. Is a glucopyranose. Now the pyranose, when you hear pyranose, you're going to think six-membered ring. We've got that prefix pyran with like pyridine a little bit, a six-membered ring. Doesn't really work, but a pyranose is a, just in general, this would be a pyran. And that's in contrast to a furan. And these are five-membered rings. And five-membered rings for furan, what that's derived from is right here. Okay. I think that's pyran. I think maybe pyran's a little bit more aromatic. I'm just going to check that really quick. Ooh, I'm glad I checked. I just was thinking about it. I'm like, gosh, I think I need some double bonds in there. So there's one pyran. You can move the double bonds if you need to. Ooh, I'm glad I checked. Thank you for being patient with me. Okay. So now we've got furan. Furan's this aromatic molecule now, a five-membered ring. Now, where do we get these? We can make five-membered ring sugars, actually. From fructose or fructose, depending on how you would like to pronounce it. So fructose is a ketone, a keto sugar, as opposed to an aldol or an aldehyde sugar. So the keto sugar, if we consider its open chain form, is going to have a CH2OH at the top and a CH2OH at the bottom. And then fructose is ultimately derived from glucose. So we're going to have this same pattern of stereocenters, except the second stereocenter, we're going to have a ketone and then nothing on the right, because that would violate the octet, which I just about did. Now, in that case, what we're going to do is allow this, and again, I said this, we're going to allow one of the OHs, and it's the OH second from the bottom, that's going to wrap around and add to the ketone and transfer its hydrogen here. And that's going to make a five-membered ring. So we have OH, OH, H, OH, H, H. Okay. So that's going to be a five-membered ring. Now that's an atrocious drawing. And so what we have to do is consider then a ring closed form. And so what the ring closed form looks like is this. Now we have all sorts of wedges and dashes. I'm going to put a squiggle here. Okay. We have OH, I'm doing the wedge dash form. I crossed my wires with a different way of representing these. So here the OH is a wedge, the OH is a dash, and then a CH2OH is a wedge. Okay, so now this is a fructofuranose. And we see these five-membered rings in nucleotides and like adenosine-like things where we have ribose present. We'll get to that here in a second. But a different depiction is the Hayworth projection, where wedges are lines up. Did I spell that right? Hayworth projection. I'm going to check on that spelling. Wedges are lines up and dashes lines down. Okay, just checking this really quick. Hayworth. Oh, just one W, which of course makes sense. So sorry about that. Hayworth. I'm going to cross that out so you can see what I did. I thought two W's look bad. Okay, so words are only so helpful with Hayworth projections. So what I'm going to do with my Hayworth projection is just start drawing them. So what we do is we draw the 5-membered ring. You could do this for 6-membered rings, but I would just prefer chair structures. What we're going to do is draw the, make the lower 2 carbons a wedge. And then instead of having wedges now added for the substituents, I'm just going to replace the wedges with an up substituent if it was a wedge, and a down substituent if it was a dash. And so, and I'm going to ignore the H's. Okay. Now these two are still squiggles, and if you're wondering why, it's because this carbon is the anomeric carbon, and so it can scramble. Now between these two species, you can get a ratio of the alpha and the beta forms. So the beta form, beta fructose, is dominant. And here it's less, my alpha axial is less helpful, but you could remember that beta, the OH is up, whereas the axial is down in the typical depiction that I have of glucose. So here, beta fructose means the OH is up. So what our Hayworth projection looks like is this. I'm not going to make the shading too thick this time, just a little bit of a splash of solid lines. Now the OH is going to be up, and then it goes up, up, down, up, CH2, OH, and then we can't forget this substituent down, which is the other CH2OH. So this is all because we have a keto form. And again, this is beta fructose, and this is a fructofuranose. And it's a furanose because it's a sugar. There's the ose suffix derived from with a 5-membered ring, which comes from furan, which is that 5-membered aromatic ring. Okay. Now, as I'm sure many of you have heard, we can remove oxygen atoms from these things, and then we get to deoxy sugars. So if you have deoxy ribose, let's first consider that as a Fischer projection. Deoxy ribose looks like this. OH, OH, CH2OH. So I'm showing the open form, and that's not a very popular form for ribose. Okay, so what we do with ribose is we first of all notice that we've got 1, 2, 3, 4, 5 carbon atoms present. And so because we have an aldehyde at the outset, we do have an opportunity to form a 6-membered ring, and that would connect this OH from here, and that would get you to a 6 ring. But and that's not a bad anomer to have. So you would have, I think it says here, it looks like you get about 23% of that 5-membered ring versus 75% of that 6-membered ring. But if we connect instead at C4 to the carbonyl, which is on the aldehyde, we make the 5-membered ring. And the 5-membered ring is where we see all of the applications. And the open chain form can fill in the rest there. Okay, so the 5-membered ring is where we see the major applications. So ribose is this 5-carbon aldose, 5-carbon aldehyde derived sugar, and this is deoxyribose because we don't have an oxygen at C2. So it's deoxy, whereas ribose would have a sugar at that position. Okay, so if we form a ribofuranose, we could have our Hayworth projection. Furanose implies the 5-membered ring formation. And we could have an anomeric carbon here. And the book has the alpha, which is down. This center is deoxygenated, so there's our deoxy center. Going left, we've got OH down, then CH2OH. Okay. Looking through this, there is always more to talk about carbohydrates, and it's all wonderful chemistry. You know, as you're copying down the last bit of notes, what I'll just mention is that what I've observed over the years from the biochemistry community is there are so many examples of great science and great scientists in the arena of carbohydrate chemistry, but it doesn't seem to get the same attention as proteins do, and amino acids, which will be the topic of the next class of biomolecules. And the proteins and the amino acids are very interesting and very exciting and do so many things. Sometimes I wonder, I'm probably just completely wrong about this, if some of the great biochemists in the protein sciences wouldn't have gotten into carbohydrates if carbohydrates were easier to kind of work with and understand. They're difficult to work with in the lab, and they're difficult to understand because structurally they have very complicated structures that are really important to understand. Like Hayworth projections are not easy to kind of think through. Fischer projections are helpful for organizing a huge class of carbohydrates, but they're not easy to think through. So anyway, as you're thinking about your future as a scientist, recognize that while we talk a lot about the opportunities available in enzyme science, the involvement of sugars and saccharides is very prevalent, and it's a very important science that should be looked at. Anyway, said my piece. We'll move on from here into more carbohydrate chemistry. I'll see you then.